first thing I thought about is that I would try to emphasize the cross-species perspective, which is so critical to our work in general, in neuroscience in general, in social neuroscience, in developmental social neuroscience, and at any point that I could connect to dialogue my work with those who spoke before me, I would do it. We work in the human species, which, as you heard, is a pretty complex species to work with, but this is what we're interested in. And but because, to quote Wittgenstein, the best model for a cat is a cat, and preferably the same cat. What we do is very long-term research on the same human. So I will present to you studies, most of the studies, or I would say maybe all the studies I present to you is longitudinal on the same cat, whatever we can make out of it. In research spanning from 4-year longitudinal studies, I think that this would be the shortest, to 25-year longitudinal study of birth to 25 years. So let us look at human evolution, not only from bacteria to mice, to primates, to humans, but also in the life of a single human or family from birth to adulthood. So let me start with these images from the work of Louise Bourgeois, a French-American artist who was preoccupied with the maternal body. And I think the marvel that we come out of someone else's body and what a profound impact this fact has on our brain and development. What does it mean to be born a mammal? Mammalian young are born with two important constraints. Their brain is immature at birth, and young maintain close contact to a nursing mother. This opens an interface in both space and time where the young, immature brain develops in the context of the stimuli that are provided to it by the mother. And here we have two types of mechanism. Mammalian general mechanism that's related to being close to the mother's body, and this is the work of Myron Hoffer beginning in the 1960s, but then many others showing that okay, let's see. Better? So here, I think this is the concept that you, Pat, mentioned is allostasis, is the kind of important that the mother's body has on all these systems that maintain energy, like autonomic maturation, motor organization, temperature regulation, sleep organization, etc. And here is the human-specific mechanism, which I am interested in, is biobehavioral synchrony. And biobehavioral synchrony is a mechanism that is defined as the coordination of biology and behavior between mother and infant during moments of social contact. So we found that during moments of synchrony, when mother and 2 or 3 months old infant coordinate their behavior, their non-verbal behavior in the gaze, affect, vocal, and touch modality, there's also coordination between the mother and infant heart rhythms, which is not coordinated during moments of no synchrony. There's a coordinated release of oxytocin, and there is brain-to-brain synchrony during these synchronous moments of brain rhythms in key nodes of the social brain, which means that behavioral synchrony, it's a bottom-up process. The behavioral synchrony provides the template for the coordination of physiological, biological systems between mother and child. What does it do, biobehavioral synchrony? And it does two things. First, lengthwise, and then widthwise. Here is Maya. This is a 25-year-old study. You see Maya from birth to 22 years. And what we see is that the coordination of nonverbal behavior between Maya and her mother expands like cycle in the pond to synchrony that has more and more abilities, more and more complexities as new cognitive abilities kick in. So you see that nonverbal synchrony in the first months of life during the sensitive period of the nonverbal communication becomes then the joint symbolic play in toddlerhood, the co-construction of narrative in preschool, the ability to see multiple perspectives in adolescence, to the young adult interacting with a mother that has intimacy, self-disclosure, and the ability for empathy. But they're all built on the same pattern. And here I'd like to give you an example from Hannah and Abigail, who was my #1 subject in my PhD research. So you'll see them first at 3 months on nonverbal synchrony, then you'll see them at 13 years, and then you'll see them at 24 years. This was about a minute and a half. This is the ability over 3 months old to interact, the episode of maintained engagement. And then the child gaze averts and relaxes, and the mother no longer interacts. But you see that this is a kind of high energy, high positive arousal, gaze synchrony between mother and child. And this is maintained when the child is 14 and they dialogue a conflict.
No, mommy, no. זה חייב להיות, את לא יכולה להיות כזאת שאת פוחדת לגדוע בכאב. לא. את תעלי. חוץ. את יודעת מה יקרה?
And here they are when Hannah is 24, same rhythms and arousal.
There's pretty places in London.
Okay, so
I think we have to settle over
here. I thought you don't like London.
I like London. It was just like, I kept expecting like Janie to pop out of a cake.
Okay, so we go to London.
Okay.
And we go to Oxford then.
Okay.
Okay, so you get the picture. And what you see, the way I conceptualize is that it's almost like the 12-bar blues that plays in the left hand, and it gives the predictability that Tali Baram was talking to us about, this predictability that's made of arousal and positive affect and gaze synchrony, and that plays in the background, and on top of it, more and more abilities kick in, and that echoes what Stu West, the evolutionary biology, talks about, is that evolution happens when the same social patterns with a new cognitive ability. When new cognitive abilities kick in the same social pattern, there is an evolutionary transition. But the widthwise is also important because when we see there is a behavioral, autonomic, hormonal, and brain synchrony, and we found them in all 4 human relationships, between parents and infants, between romantic partners, between close friends, and in human. Unlike other mammals, humans can also synchronize their behavior and affiliative biology with those of total strangers. Just one word about the system that underpins these behavior, the oxytocin system. We heard about it extensively from Larry, but just to put it in evolutionary perspective, the oxytocin system is actually a very ancient system. It is presumably evolved via gene duplication in jawed fish approximately 600 million years ago, and it played a role in the regulation, in allostasis, in the regulation of basic life functions such as energy conservation or water balance or thermoregulation. With the evolution of mammals, those functions were repurposed and became attached to the attachment context. So oxytocin became involved in maternal-infant bonding, in pair bonding, in social hierarchies. So within the parent-infant relationship, this is the place where the oxytocin system now operates, and all those regulatory functions are acquired. So this is how I would do my talk. I'll first talk about the brain basis of human mothering and fathering, and then about the long-term implication of the human parental brain. I will then ask, how do relationships build brain? What are the long-term implications of parenting to the development of children's social brain? I would first bring example from typically developing children, but I would use two models that we've been studying in our lab for about 2 decades, one of maternal postpartum depression and one of premature infants. Both are models to impairment or break in maternal-infant bonding. And then I thought we could all discuss about the conceptual issues, and if we have some time, the clinical implications of the finding. So this is a figure from Larry Young's paper on the maternal brain in rodents. Interest in the rodent maternal brain is about a century old, although human imaging studies are also only about maybe 15 years or so. There's a central role of the MPOA, an area in the hypothalamus, primed by hormones of pregnancy, project to the VTA, maternal reward from the infant, to the amygdala, maternal vigilance, and together the three of them create this subcortical network that support parenting in mammals. There is a similar network that activates in father, although they are orchestrated by different neurons. Like new neurochemical studies show that different neurons are responsible for mothering and fathering in the same structures. But animal research paid very little attention to cortical structures and their role in parenting. And what we see here, what I put here at that side, is an enlarged view of the striatum and all the associated area that are implicated in the mammalian parental caregiving network. So here's what you see in rodents, but in the middle here, you see it embedded within the human brain. So what you see in the human brain, that it is there, it is critical for parenting, but it is connected via multiple ascending and descending projection to several very, very important cortical areas that are implicated in human parenting. And this is a meta-analysis that we did on the basis of about 50 published studies of the human parental brain. We see in them the subcortical network, primarily including the hypothalamus, oxytocin-producing hypothalamus, the amygdala and the VTA, the striatal areas of the dopamine, subcortical dopaminergic circuit, and they are connected to 3 important cortical networks. The embodied simulation network, I think Valeria and Christian told us about us. This is a network that enables the parent to represent the infant's action or emotion in their own brain. The mentalizing network, mainly temporal or temporal-parietal areas that enable parents to predict infant intention from their action. This is a cry of hunger, this is a cry of tired, etc. And the latest evolving emotion regulation network that enable, like Jean say, to put valuation, to plan, to multitask, to plan long-term parenting to that child in relation to culture, to context, to belief system. So in human, all this is orchestrated, evolved on top of the mammalian subcortical caregiving network. So in parenting, we're very similar to other species, to other mammals, that enable us to really use cross-species comparison, but also not to forget the human-specific aspects of parenting that really enable us to provide parenting that is mindful and connected to our culture and our values. This is the type of research we do on the parental brain, and we've done several studies of the maternal, paternal, co-parental brain, and just Lawrence's work we use ethologically valid stimuli. Just like Lawrence went to the pond to look at the duckling, we go to the home, we film the infant, we film the parent-infant interaction, we film triadic interaction between the two parent and the child, we film the infant alone, we film the parent alone doing dishes or reading newspaper. We use those ethologically valid stimuli and compare one's own baby or one's own interaction with a baby with unfamiliar interactions. So we want to see what is it about attachment, about the exclusivity of the infant's own attachment, what are the networks that activate when you watch your own infant or when you watch interaction with your own infant. And we use video clips, not, we were basically the first ones, I think, in Shear's study. Shear's here, he was here before. This was the first study that that was used video clips, not just unimodal, but we want to create as ecologically valid stimuli as possible. And so we compare 2-minute clip of own interaction, unfamiliar interaction, own infant, unfamiliar infant, various things. So let me describe to you one of our studies of the parental brain, and there are 2 reasons why I chose this study to describe. First of all, because it gives you the brains of mothers and fathers, and it distinguishes between mothers being in the primary caregiving role to father being in the primary caregiving role, and it also gives us biological parents versus adoptive parents. So it gives you a viewpoint of all types of parenting to a few months old infant. And the second reason I'm using this study is because this study we followed these children for now 10 years, and I have data to show you for the first 6 years. So this is a longitudinal study. It is the largest sample of the parental brain today. We had 91 first-time parents, including 48 homosexual fathers. So we had pairs of gay fathers that were in a committed relationship. A single parent raising a child, it's already a whole different type of brain. It's much more stress. And they had the child via surrogacy, so the child was raised without any maternal input from birth, but there was no maternal separation, so that the minute this child was born, it was given to these 2 fathers, okay? In each gay couple, there was one biological parent and one adoptive parent. And they were compared to first-time mother and father. And we had extensive interviews showing who was a primary caregiver, who was secondary caregiver. So in the heterosexual couples, mother was primary caregiver, father was secondary caregiver, and in the gay fathers, they were both primary caregiver by all accounts. We went to their homes, and we collected saliva samples for hormones, and we collected the interactions. We coded the interaction for parent-child synchrony, and we used those interactions we filmed in the home in the magnet a week or 2 later. ככה? אוקיי. נראה לך, זה עכשיו שומעים יותר טוב? כן. תודה. מה? אני מעדיף. בסדר? עכשיו יותר טוב? לא, זה בסדר. עכשיו אתם שומעים טוב? Do you hear better? Okay. No, wrong side. Okay, and what we found that across parent type, mothers and fathers, primary caregivers and secondary caregivers, and biological parents and adoptive parents, basically most of the nodes of the global human caregiving system were activated to the same degree. So we have the subcortical mammalian network, we have the embodied simulation network, and we have the mentalizing network, and you see here also areas in the emotion regulation network. So mainly we see that all types of parents, when you raise a child and you assume the role of a caregiver, this activates the human caregiving system. And think about it is that until like 150 years ago, every third mother died at childbirth. And many children were raised by women who were non-healed. So there must be a system for us to survive as a species. There must be a system in the parental brain, in the adult brain that flexibly activates when you take upon yourself the role of taking care of an infant and you assume caregiving behavior. However, there were two areas that showed significant differences between the groups. The amygdala, you see, had 5 times greater activity in mothers compared to fathers, and the STS, which is a key node of the mentalizing network, actually it's a node that integrates mental and mirror or mental and embodied simulation properties, much more in, like 3.5 times more in fathers than in mothers, and the amygdala is pretty understandable. We look at it as a more evolutionary ancient pathway to mothering. Mothers undergo pregnancy and childbirth. There are a lot of oxytocin receptors in the amygdala. Amygdala is sensitized across all mammals by pregnancy and labor. So this activates the amygdala. And the SCS, mentalizing, fathers take care of the infants and they have a more cortical pathway to the human parental brain. But what was really interesting is that primary caregiving fathers had amygdala activation like mother and obviously STS activation like father. But this is interesting. How do those fathers who did not go through pregnancy and childbirth had amygdala activation that's 5 times greater than a non-pregnant mother or a father? So we first look at correlation between hormones and brain activation or synchronous behavior and brain activation, and we did not find. With fathers, both primary caregivers and secondary caregivers, father's STS was related to both parent-child synchronous behavior and parent-child oxytocin, and mothers were different. But then we found that only for the primary caregiving fathers group, there was a functional connectivity between the amygdala and STS. And you couldn't find this connectivity in the other groups of secondary caregiving fathers or mothers, as if the paternal pathway recruits the maternal pathway to preserve infant from harm when mothers are not present. Now, once we saw this mechanism of connectivity between the amygdala and the STS, we looked at it across all fathers, and we found that the degree of childcare activity the father performed alone to the child, the amount of time and the amount of caregiving activity. It's not a father who comes home and plays with a child many hours after the child has been fed and bathed, but the father was really active in doing every caregiving activity. The more they engaged in those activities alone, without the mother, the more the STS in that father was connected to the amygdala. So what we say, maybe as a general statement, is that mothers get the caregiving system activated for free. It's really not for free. They carried the infant for 9 months, and then they went through labor, and that activates the system. Obviously, many things can go afterwards. You'll see with the depressed mothers, and the kid could be premature, and things can go, but if everything is healthy, mother's brain is prepared to receive the infant. Fathers don't get it for free. You work, you'll get it, right? You'll be a high-tech dad, and you won't see the child, and you won't engage in child-rearing activity, then the activation of those systems will be much reduced. Then the second question we had, and I think Ronnie had it before, or several of you speakers said it before, what do these labs mean for later child development? So we see very nice activation in the maternal, in the maternal, in the primary, in the secondary. You are biological, you aren't biological. Does it mean anything for child development, or those are very nice labs that make for very, very beautiful graphs that you could send for publication? So we wanted to see if anything about the activation of the parental brain means something for later development.
You can comment on the heterogeneity. I mean, if you look at the distribution here, and so you put, what is that heterogeneity? Is there anything that heterogeneity predicts later on?
Right, so here I am going to show you data from when the children are preschoolers and then when the children are 6 years. So this heterogeneity obviously relates to the parents.
You have the individuals here that are low on the average weekly hours alone, but high on functional connectivity, have the inverse.
Right.
I mean, minority of the whole population, but enough of them to ask, what is that? What's driving that connectivity, and what does that predict later on if they have weekly hours alone that are low?
Right. Right. So let me show you the data of the connectivity, because we went on the connectivity indices rather than on activity indices. So what we did when the children were preschoolers, between 3 and 4, we wanted to measure their social and emotional development. And in my lab, we don't usually ask individual questionnaires. We do that very little. We observe them. So we did several paradigms at home. This is a socialization paradigm. They play, and then all the toys are out, and then we ask them to pick the toys up for the next person to play with, and this is an index of morality or moral development. This is the regulation of negative emotion. This is a mask paradigm, and we microcode for both the amount of negative and positive emotion and the strategies children use to regulate moments of fear, and then a regulation of positive emotion. We use a bubble play, a soap bubble play, and we ask again what is the amount of positive emotion and what they use to regulate it. And here we use NCI, Network Cohesiveness Indices, and what these indices tell us is how areas in a network stick together, function together. So it's a functional connectivity in the main. It's a functional connectivity index of areas in the network. So these are areas in the subcortical limbic network, in the embodied simulation network, and in the mentalizing network. And the NCI index asks how well they connect together as a network within themselves, and then there is an intra-NCI, which asks the question of how well two networks connect together. So here are the results for 3-year-old based on the parents' brain organization, cohesiveness of these networks in the first months of life. So what we saw is that parents who had greater connectivity or cohesiveness of the subcortical caregiving network, the mammalian general, had more positive arousal expression. They were laughing more during the positive vignette, and they used simple mammalian general mechanism to regulate moments of heightened arousal, like proximity seeking. When they were scared, they went to the parent. When they were laughing, they wanted a hug. Embodied simulation, parents who had greater connectivity of the embodied simulation network had children who were better able to use complex regulatory mechanisms to regulate moments of intense negative arousal, moments of fear. Like they used attention diversion or symbolic play. They didn't cry or decompose, but they used more complex mechanisms to regulate negative emotion. And parents who had greater connectivity of the mentalizing network were children who were better socialized. Interestingly, these children at 4 years old, children of parents who had better connectivity of the embodied simulation network had lower level of cortisol response to the fear paradigm, and also the link between embodied simulation, NCI, and the child use of complex regulatory behavior towards a stranger in a negative emotionality paradigm was mediated by the degree of parent-infant synchrony. So we think that parent-infant synchrony really tunes the child brain to the social world and enables children to use more complex mechanism when they meet various events or emotionally laden episodes in the environment. What we see at 6 years of age is that the connectivity between these two cortical networks, the embodied simulation and the mentalizing network, actually those two networks are thought to underpin empathy, emotional empathy, and empathy, respectively. So better connectivity in the parent's brain. Children at 6 years of age had lower internalizing behavior, and this was mediated by regulatory behavior of the child at 4 years of age. So here you see that activation of the parent's brain to the child, which is really the interface of where mammals raise their children. An important, perhaps the most important mammalian brain alteration that occurs and takes care of the child is meaningful to how that child is going to develop socially, probably because of its impact on parental brains. So here we come to the next part of my talk is that how do relationships build brain? So the first, what I showed you with the first sample is how the brain of the parent builds the relational, the social and emotional repertoire of the child, and here we'll do the other way around in the next 3 studies. How do relationship patterns between parent and child build the child brain? Okay, we still don't have, we're working on it now, relationship between the parental brain right after the child is born to the child's brain later on. Okay, so it's right now it's in 2 pieces. This is May study, and you see Yoni. Who's Yoni? Hi, you see Yoni, and Yoni read that study. Yoni and May. So this is a study where we videotape mother and child. Children were between 8 and 9 years old. Again, we videotape mother and child in their home environment in 2 paradigms, as we do always at this age. There was a positive paradigm when they had to talk about a positive theme. They had to plan a fun day together. And then they had a conflict discussion. We asked them to be the relationship and discuss it. And then we coded the conflict for various global measures like empathy and reciprocity, that would be the interaction. And the positive interaction, like we did in infancy, was micro-communicated second by second for gaze and affect symptoms. And then we used this interaction and showed it to the mother in the neck and to the child in the neck. So they were exposed to exactly the same video clip. Mother is watching, mother-child interaction versus unfamiliar interaction, and the child is exposed to the same interaction. So it's not yet real life brain-to-brain synchrony. It's not like we're measuring their brain while they're interacting, but we're exposing them to exactly the same thing. Typically in the same day, 1.5 weeks. And then we might suppose positive, mother and child observe their own versus unfamiliar
interaction in the neck. We computed brain oscillation and synchrony between the brain oscillatory patterns of mother and child in response to viewing themselves versus unruly interaction. What we found is that in general, observing your own interaction in both mother and child activated the STS. So you had greater activation in the STS to your own interaction versus unfamiliar interaction in both mother and individual child. We also found that they were coupled between each other, so that the activation in the mother's STS and in the child STS were coupled, and that it was in gamma rhythm. Now, gamma is very important, and I'll talk about it in a minute. But the really nice thing is that gamma synchrony between mother's and child brain were only observed during moments of behavioral synchrony. So here you see when mother and child synchronized their behavior in the gaze and affect modality. They were looking at each other, they were smiling together, they were co-vocalizing. During those moments, there was brain-to-brain synchrony of gamma rhythms in the STS. And during moments, a second later, when they weren't synchronized, and the brain of those mother and child were not synchronized. Now, gamma is a very important mechanism. We then went on to see in real hyperscanning interaction. That's a mere study. He'll describe it tomorrow in couples and in stranger during real life interaction, both wearing EEG cap, and we found the same. Couples, but not stranger, synchronize their gamma rhythm in temporal cortex. E.g., we really can't localize it so exactly to the STS, but it's pretty much the same area. And it's, again, in moments when the couples coordinated their behavior, but not during moments when behavior wasn't synchronized. We think that gamma, which in the predictive coding frame is a rhythm where you compare incoming information to your prior prediction. So it's a rhythm that charts maturity. It appears in animals only as a signal of maturation late in development. I think, Pat, you said that the children with the ACEs were not showing gamma rhythms. Lower gamma rhythms. And we see that mothers who live in Sderot, in our war-exposed mothers who are exposed to war, when they are asked to empathize, paradigms of empathy, they do not show gamma rhythms, whereas the mothers who do not live at the border do show gamma rhythms. So gamma is probably a mechanism for parenting, for understanding your child, for comparing what the child wants, but it's also a mechanism for brain-to-brain synchrony, where two brains coordinate their behavior in relation to the synchrony that occurs between them. Okay? And now, two models, two longitudinal studies that model disruptions in maternal-infant bonding, and let's see what it does to parent-infant behavioral synchrony, what it does to the child's brain, and what it does to the oxytocin system. So here we wanted to do, Moshe, you would like it, we wanted to follow Michael Rini and Moshe's study, very elegant animal researchers. So you remember the licking and grooming study in rat moms. So they took a large group of rat moms and they observed who were the high licker and groomer, the low licker and groomer, natural variation in the population. Some mothers just naturally lick and groom more, some naturally lick and groom less. They took the two extremes, they bred them for several generations, and we have two groups, mothers who were high licking and grooming, mothers who were low licking and grooming, and then they did all the brain and neurochemistry tests on the two groups to show how they differ. And this is how we conceptualize maternal depression, as low licking and grooming mothers. So this is what we wanted to do, and we wanted to get a very clean samples. We did not want maternal depression to be confounded like it always does with poverty and single parenting and premature birth and teenage parenting, or in the 35% cases where depression is confounded with anxiety, because at least for the oxytocin system, depression is associated with suppression of the oxytocin system, and anxiety may be associated at times with hyperactivation of the oxytocin system. So this is what we did. We went to 3 hospitals in the Tel Aviv area, and we recruited 1,983, it's not a year, it's a number, mothers who were middle class, above 21, no poverty, with a husband, high school education, healthy, the child is healthy, no preemie, no tweens, etc. And they reported on their depression and anxiety. At 6 months, we used those who received lower on depression and those who were higher on depression, and we excluded mothers with high anxiety. In 9 months, we did the same. 254 mothers, excluding those with lower depression, and using only those in the lower quartile and those at the higher quartile, and excluding those with high anxiety. And of the 254 mothers, we called them all, and we went to visit the homes of 192 mothers at home visit, and we did a clinical diagnosis of those mothers who were clinically depressed, and we observed the mother-child interaction, we collected hormones, etc. At 6 years, we were left, this was not a choice anymore, this was a general attrition, with 156 mothers and fathers. We went again to the home, we collected DNA and interaction and hormone and measures of empathy and neurocognitive measures, and we came up with 2 groups that are parallel to the high lickers and the low lickers. So we had 103 healthy mothers. We excluded all those who were in the middle, or all those who received other psychiatric diagnoses. So we had 103 healthy mothers who reported no depression at birth, 6 and 9 months, and were not diagnosed with any psychiatric disorder at 9 months or 6 years, and reported being relatively healthy and happy throughout the child's first years of life. And then we had the depressed group, and this depressed group is especially depressed group. They received high score on the BDI at birth, not 6 months and 9 months. They were diagnosed with unipolar depression at 9 months, and again diagnosed with unipolar depression at 6 years, and reported being relatively depressed. When depression is a cyclic disorder, relatively depressed throughout most of the child's 6 years of life. So we had 46 women out of this 1,983 women we initially recruited, but those were our very special groups, mothers who were depressed throughout all the early childhood, were not anxious, and were of middle-class, low-risk contextual background. So the child had a father, and everything was good financially around it, lived in a safe neighborhood, etc. But the mother was depressed throughout the most of the child years, and then we had 135 mothers and children in a home visit, and children were about 9 and 10 years. And then we had a neck scan study of 92 children, when children were about 12 at pre-adolescence. And from all this immense data, I will show you only the lines that matter. Mother-infant synchrony at 9 months, the child oxytocin at 6 years, and the child's brain response to attachment cue. This is the first study where we use children's response to own versus other interaction. So it's the child watching his interaction with the mother versus unfamiliar interaction with the mother. So what we see in a last sequential analysis that much less synchrony in depressed mothers than in typical mother, but the way it looks is that when the infant positive vocalizes laugh to the mothers within 2 minutes, this is a last sequential analysis, mothers will gaze avert, as if they're not able to contain it or be in a relationship or an interaction, visual interaction with a laughing infant. After a little bit, the child will start being positive. And the same thing when the child gazes at the mother. So a depressed mother terminates the child's beautiful sort of interaction. This is what we see. And then they get into a cycle of, I think Tali will call it not only unpredictability, but also ahedonia. Because then the interaction becomes very stable, and it takes depressed mothers 5 times longer to initiate the first eye contact, 60 seconds versus 12 seconds in mother, and the cycle between one gaze synchrony to the next takes 7 times longer. So those are very, very micro level processes, but they take a very, very different rhythm and dynamic from Hannah and Abigail you saw on the first slides. This is what you see when the children are 6 years. 60% of the children, by the time they enter school, receive a clinical diagnosis. This is staggering. But again, this is not your typically depressed mother that is depressed for a little bit and then remits. These are mothers who have been relatively stably depressed for the child's first years of life. This is 4 times more than approximately 14% of children to well mothers receive a clinical diagnosis. And this is in relation to the oxytocin. We see, this is a salivary oxytocin, but we see it in urine also. We have much lower oxytocin level in the mother, in the child, and in the father also. And it's not clear, none of our father had any psychiatric diagnosis, so it's either assortative mating, birds of a feather stick together, and oxytocin is relatively stable. Mothers who are low oxytocin probably choose low oxytocin men, or in some kind of biobehavioral synchrony that the low oxytocin level in the mother somehow impacts the father, but the children are growing in low oxytocin environment. And this is what you see in the child's brain, and you see it again in beta rhythms and gamma rhythms, which is what you showed with much lower activations in children who were exposed to early life stress. And what we're beginning to think now as these children are growing older, we just had a paper showing much more oxytocin, but impairment in the immune system, that being raised with a chronically depressed mother is it's very specific early life stress. It's like it activates your immune system. It's like growing up in a disease condition. It's something that the brain and the body have to imagine. So what you see here, and you see it in the children of depressed mother, not only lower activation of beta and gamma to own versus unfamiliar interaction, but also no differentiation. So if we're talking about internal working model of attachment, the reward system that Larry is speaking about that connection between oxytocin, which is socially directing us to our attachment, and the reward. So here you see, and then again, this is the areas. Gamma is a wider, more spread, and beta, they're basically the same area of the posterior STS and the right insula, where the children are not activating specifically or exclusively to attachment stimuli, and this probably would have implication for their later attachment life. But what is interesting is has to do with resilience, and we'll talk about it in relation to resilience. This was not this pattern wasn't observed, and I think that's back to your question, Pat. This wasn't observed in all children of depressed mothers, only in those who developed
the 12 bowel
disorder themselves. Children of depressed mothers who had better oxytocin, better allele on the oxytocin receptor gene 298, had the less risky allele, the A allele. If the mother had better oxytocin system, the child's propensity to develop a psychiatric disorder was reduced by half. Not 60%, by 30%. 30% is still bad, but it was reduced by half. If the mother, if the child did not develop an affective disorder, this pattern was much less severe. So there are individual differences. The oxytocin system does confer resilience in the context of maternal depression. How we can use it to our benefit is still a question, but not all children are impacted by chronic maternal depression to the same extent, and variability on the oxytocin system. So maybe if we can give children during the sensitive period some oxytocin-producing experiences like touch or hug or gay synchrony, maybe we can somehow alter the trajectory. And oxytocin level at 6 years predicted beta rhythms to both own interaction and unfamiliar interaction, and we think of it, just like Larry said, as a general socially orienting. Oxytocin system orients you to social interaction, and then it's fun to read later. So oxytocin orients you to attend to social interactions.
Some of the variability maybe also be explained in terms of the degree of the depression, the childhood depression in the kids of mothers who are depressed, be explained by the other partners in interaction. In other words, I didn't see anything measuring how involved the other caretaker was, since it doesn't have to be the mother to get the oxytocin system going.
So I apologize, this is a story I didn't tell you, but there are 3 papers that are out already. Fathers can mitigate many of the effects, but it's a story I didn't lack of time. But some of the variability has to do with what the child, and actually involved sensitive synchronous fathering has a much greater effect on the development of children of depressed mothers than in children of well mothers. Okay. So if you don't have one and you have the other one better, there are compensatory mechanisms, not on all systems to the same extent, but I'm sorry, there's like a huge amount of data for this study. Okay, and let's quickly end with our last sample. This is a 20-year sample. Kangaroo here. This is the daughter of my best friend. Actually, my best friend. Me and Odeya's mother are friends from 3 years of age. And we went to kindergarten together. And at the time, we were just getting the brain results. Her child had a preemie, like a real preemie, a 1,000 gram preemie. And I told her up and I said, I'm coming to show you the data. Kangaroo, kangaroo, kangaroo. So this is Odeya with David and their kangaroo. So this is our study. It started in 96. The children are born between 96 and 99. Before kangaroo here became common knowledge. You can't do that anymore. You can't randomize, because touch and contact is not something you are ethically allowed to tell. This is where human studies differ from animal studies. You can't randomize. But the way we did it was, because at that time kangaroo here wasn't known, we introduced them at 2 hospitals and we did a crossover in order for the kangaroo care control to be exactly measured on all parameters, demographic and medical parameters. We targeted kangaroo care to the sensitive period when mothers are or aren't able to hold the child fully. Mothers had to sign to be in our program to be willing to do kangaroo care for at least an hour a day for at least 14 consecutive days. And we had all these observations. Pregnant kangaroo at 37 weeks gestation age, usually it's the time when mother and infant leave the hospital. 3 months, 6 months, 12 months, and 24 months corrected age is Aaron here. Aaron was collaborating with us on that project. And then after 24 months, you no longer count until gestation. You no longer do corrected age. 5 years, 10 years, and 18 to 20 years. They were imaged by D. A D imaged the children at 18 to 20 years. And we took a bunch of measures, mother-child interaction at all times, father-child interaction at one time, emotion regulation paradigms at each age, hormonal collection, physiological processes, cognitive assessment, and 18 to 20 years. We image them, and I'll show you just one paradigm, and you see the results also in these poster. So what you see is that at each time point, and this is a question I think that Moshe raised, how does this intervention, providing maternal infant skin-to-skin contact during this sensitive period when a child needs to be held by the mother and isn't or is for 2 weeks only, what does it do? Does it tip the child's trajectory to a more optimal trajectory? I think this is what it does, that the child's regulatory systems become a bit better, they elicit better caregiving interacts with more emotion regulation, and they influence each other over time until they make a very big physiological and brain impact. So at 3 months, 6 months, 12 months, 24 months, 5 years, 10 years, and 20 years, we see greater synchrony, empathy, reciprocity in the mother-child interaction between the kangaroo here and the controlled children. I just want you to realize that these are healthy children. We specifically selected preemies that could develop healthy, and when they come to the lab, they're big. Like you see these 18-year-olds, you don't know that they're 600 grams, and they all look fine. So we're talking about differences that are very subtle, okay? But the mother-child interaction across development is better. Here we see 2 hormonal measures. There is greater cortisol in both mother and child, and it has to do with the early experience of not connecting to that child. This is area under the curve. I think the home visit was about 2 hours. So we collected several salivary samples from the mother and the child, and we measured area under the curve. And this is an immune biomarker, and in the immune biomarker SIGA and the mucosal barrier, the first mucosal barrier, and you see that here is just for the child, and again, telling you that being born prematurely and not having contact with the mother is an early life stress, and it activates the immune system that you can see it 20 years later, and that kangaroo here rescues it. And this is the brain paradigm that Adi did in the scanner. This is an empathy paradigm. We ask if you can imagine, it's a paradigm from Silvia Morelli at UCLA, and she asks, please put yourself in someone else's shoes, and there are 3 emotional contexts, sadness, anxiety, and happiness. This person lost an important sport competition. This person might be fired soon. This person won a competition, whatever. There's a neutral condition. This person is tying his shoes, and then we see the scramble condition. So after each sentence, they see 4 pictures in the magnet that describe what we just see. So these posters tell you about the amygdala and the medial prefrontal cortex diminished activation in the control and increased activation. But I wanted to focus on a different system, and this is the supportive overview reward system. So here you see very, very different. Actually, this is really interesting because you see midbrain activation in the kangaroo here, which in the other group you don't see at all, and that's really interesting. Like you don't see any of it. And here you see very marked differences in VTA activation in the neutral versus active. So you have to put yourself in the shoes of someone else's blue hat. They won a competition. Something terrific just happened to them. So a child who received maternal contact 20 years before is able to use that support for maternal caregiving network, dopamine release, to put himself in someone else's shoes, and the other one isn't. But the other one has compensatory cortical mechanism, and they activate the STS significantly more. So this is perhaps something to do with maternal contact in the early sensitive period that enables you to really activate these unconscious and cortical rewarding experiences. So as far as self-reports or as far as behaving in the world, they may both be behaving just as empathically, but that the underlying coloring behind it is that of dopamine and cortical activation. I find that to be this preliminary results. We still have to look at the network, we still have to see what it connects to, but I think this is like a very telling that we begin with maternal brain activation, activity in the maternal brain with the VTA being a very, very key structure. Okay? And here we see the 20 years later, the provision of maternal care during the early period helps children face the world with their dopamine intact. A word about conceptual implication. I was thinking about Gerald Edelman's and the question of the subjectivity gap. Gerald Edelman, who is, I think, way over 90, right? Way over 90. He's also a mayor, he's also a violinist.
Not as old as Moses, please.
Right. And he talks about the subjectivity gap, and I was thinking about it that we should be mindful for it. This is an exclusively human issue, and it's the question of how we use science to look at human experience. Looking at it from a third-person perspective, and looking and describing an experience from a first-person perspective. Because I think the question you raised, Christian, about looking at single neurons in the human brain will resolve, I would give it 10 years, will be resolved with more sophisticated machine. This is not a question that's not resolvable. This is a question that's not resolvable. How do we describe the internal experiencing of how do we describe something about the experiencing the human from the outside? Okay? This is a question that Husserl has voiced in a beautiful article called Philosophy is Rigorous Science, where he contrasted experimental psychology when you look at the behaving human versus phenomenology, which is the intentionality of the experiencing mindful human. And I don't know how to resolve it, but I think perhaps this four-pole matrix of biobehavioral where behavior coordinates to behavior, and biology coordinates with biology, with two interacting humans. And behavior creates brain synchrony, and brain synchrony creates biology, does something that perhaps can begin to formulate a language that can transcend the first to third person perspective, because when two minds, two brains act together, and you look at it, when an internal experience becomes an intersubjective experience, one thing that happens is that it also becomes available to scientific scrutiny. It's no longer a solar system experience. So I think there's a long way with brain-to-brain synchrony research, bio-brain synchrony research, but perhaps we can begin to chart the language or develop or mark the terrain where we can begin to look at what Edelman calls brain-based epistemology. And let me just end here by thank you.